Welcome! Thanks for watching this dairy video brought to you by DairyXnet. In this video, Dr. Galvão joins us to discuss the possibility of a genetic component of uterine diseases. Dr. Galvão has over 15 years of experience in the area of dairy production, having worked in the Central Valley of California and upstate New York. He completed a residency in dairy production medicine and a master's in preventive veterinary medicine at UC Davis, then a PhD in reproductive physiology with a minor in immunology at Cornell University. Dr. Galvão moved on to The Ohio State University as a clinical assistant professor before taking a tenure-track assistant professor position in food animal reproduction and medicine at University of Florida, where he is now an associate professor. His workload is divided into research, service, teaching, and extension related to dairy cow reproduction. His research focuses on the reproductive physiology and immunology of dairy cows, development of computer models to estimate profitability of different reproductive protocols, and improvement of animal health and well-being, particularly as it relates to uterine diseases. Thanks for joining us today, Dr. Galvão.
Well, thank you very much for the introduction. Let's talk about uterine health and potential connection with genetic variation today. Just as a brief outline for the presentation today, I'll introduce you to a project that we have been working on for the last 4 or 5 years, which is for genetic selection for fertility. It's a USDA-funded project. Then we'll get to effects of disease on fertility and some preliminary work that our geneticist, Dr. Francisco Peña-Garicano, has been working on for the last 2 or 3 years. And hopefully we have some take-home message to take with us today. Well, this USDA project is a large project with several institutions involved, and you have all the collaborators here, all the universities from Colorado State, University of Minnesota, Wisconsin, Cornell, the Ohio State, Texas A&M, University of Florida, and University of Illinois. Well, for this project, the specific objectives were to identify genetic markers, also called SNPs, those are single nucleotide polymorphisms, of fertility in Holstein cows, with the ultimate goal of incorporating this into the breeding pool for sire selection for improved fertility. So one of the first questions we had was, what is a fertile cow? There is no clear definition for what's a fertile cow. So some might say uterine health is very important for fertility. So a fertile cow might be a cow that never develops uterine diseases or disorders. Others might say, well, for a cow to get pregnant, she needs to first start cycling. So we might say that a cow that is fertile is a cow that starts cycling early. Others might say, well, for a cow to be fertile, she needs to show estrus, so we can inseminate this cow. So some might say that a cow that is fertile is a cow that shows strong estrus. Some might say, well, what really matters is if a cow gets pregnant. So we might say that a cow that is fertile is a cow that gets pregnant the first try. Some might come back and say, well, it doesn't help if the cow gets pregnant, but she loses the pregnancy. So you might say that a cow that is fertile is a cow that does not lose the pregnancy after it gets pregnant. So that's why we evaluated several parameters to come up with a definition of what's a fertile cow. And we wanted to actually look at several parameters, including uterine health, cyclicity, estrus, and also ultimately if they got pregnant and they maintain pregnancy. So of course, for these genetic studies, we need a large sample size, and we evaluated around 12,000 cows, around 2,400 cows per state, and 3 to 6 farms per state. And also we collected cows in the cool and hot months of the year, because we know that heat stress is one of the main factors that affects fertility in Holstein cows. So we wanted to have half of the cows collected in the cool season and half of the cows collected in the hot season. And of course, we have evaluated uterine health, knowing that that's one of the most important things that would affect fertility in Holstein cows. So we use this tool here that you can see on the right. That is a Metritis tool that we can evaluate the uterine discharge and see if these cows have uterine diseases such as metritis and clinical endometritis. We also evaluated cyclicity, so we evaluated resumption of postpartum ovulation. We also evaluate pregnancy, artificial insemination, maintenance of pregnancy, and other health disorders such as mastitis, lameness, DA, pneumonia, and so on. So here the focus today is going to be on uterine diseases. Here we have the two most common and most important ones for hosting cows, which is metritis and clinical endometritis. So metritis is this acute, severe disease that occurs shortly after calving, usually in the first 2 weeks postpartum, with a peak incidence around day 6-7 postpartum. So we see this watery, red, brownish, foul-smelling uterine discharge that can kill cows. This is a cow that died 3 days postpartum with acute metritis. And here is a person evaluating a uterine discharge just by rectal palpation, seeing if the cow has metritis. And here we see this watery, red, brownish. You cannot smell, but for sure this will be a foul smell in uterine discharge. Then cows that develop metritis have a high chance of going on to develop clinical endometritis. So this red brownish uterine discharge becomes more of a purulent discharge. So we can use the Metritis tool to evaluate the discharges coming out of the uterus into the fundus of the vagina. And if this cow has this mucopurulent discharge, we consider this cow to have clinical endometritis. So here on this slide we have the instance of disease for this large USDA-funded project. So here we see that for these uterine diseases, metritis and clinical endometritis, we have an incidence of around 22 to 25%. And this is in line with the previous literature that shows an incidence of metritis and clinical endometritis around. But most importantly, not shown here, but very important, is that there is a large variation among herds. Some herds having an incidence of this uterine disease of around 8 to 10%, and some herds having an incidence of around 30%. So definitely there are management practices that are impacting the incidence of these uterine diseases. So there is definitely room for improvement. Even 8 to 10% would be considered large, but here we have 22 to 25%. So this is a very high incidence in general in the United States. And here we also have incidence of other conditions such as calving problems, which are predisposing factors to metritis and clinical metritis. So clinical ketosis as well for these uterine disease, and other conditions such as mastitis, lameness, DA, and pneumonia, which have lower incidence. But here we can see the factors that affect the risk of pregnancy to first artificial insemination. And we have here healthy cows with a conception rate to first service of around 42%. Quite good conception rate for healthy cows. Then we see that for cows that develop uterine disease such as metritis and endometritis, a sharp drop in conception rate to first service. Metritis cows that develop metritis having a conception rate of around 33%. And cows that develop clinical endometritis having a conception rate of around 30%. So you see here a drop in 7 to 10 percentage points, which would be 20 to 25% drop in conception rate to first service. And other diseases as well having also a negative effect, such as disorders such as calving problems, and these are dystocia, twins, stillbirth, and retained placenta. Subclinical ketosis. Mastitis here did not have a large effect, but believe me, mastitis does affect the risk of pregnancy. Lameness, DA, and pneumonia. So there are no good diseases. They all affect fertility in different degrees. So this slide we have factors affecting the risk of pregnancy loss after cows get pregnant to first service. So here we have healthy cows having a pregnancy loss of around 8.5%, while cows that develop uterine disease such as metritis and clinical endometritis having pregnancy loss of around 13%. And here the only other disease that affected the risk of pregnancy loss was subclinical ketosis. and the other conditions such as calving problems, mastitis, lameness, DA, pneumonia, not having a significant effect on pregnancy loss. Again, showing the relevance of uterine diseases to fertility, in this case, pregnancy loss. So here we have the effect of metritis on culling. So you can see here that metritis not only affects fertility, but also affects the chance of cows leaving the herd. And the effect is most prominent in the first 60 days postpartum. See, here we have cows with metritis in green, and their line is dropping faster than the cows in blue, which are cows that did not develop metritis. That means these cows are either dying or being sold at a faster rate than cows that did not develop metritis. And these lines separate pretty much in the first 60 days, then they become parallel, meaning that after the 60 days, after 6 days postpartum, the chance of these cows being sold or dying becomes the same, comparing the cows that develop metritis and cows that did not develop metritis. Well, you probably know, but metritis also affects milk production, and in general, the loss that we see in the first 30 to 60 days, about 5 kilograms per day, which is around 12 pounds. So it's a lot of milk that we're losing from these cows that develop metritis. So one question we had was that what happens to a cow that develops metritis? This is an infectious disease, and well, we have the immune system fighting this disease and the cow gets better. So usually when that happens, cows acquire some level of immunity to that disease. That's why we vaccinate cattle, because we want to have them exposed to the pathogens that cause that disease, so the next time that the cows encounter that pathogen, they have memory. They can fight off that disease faster. So that's one possibility, that cows that get metritis will be less likely to develop metritis in a subsequent lactation. The other alternative is that actually cows develop metritis because they have something intrinsic to them. For example, in their genome, their genetics predisposes them to metritis. So we use this large data set from one of our local farms to ask that question. So here we categorize cows as having metritis in one lactation, then looking at what happens to them in the subsequent lactation. So we created pairs comparing one lactation with a subsequent lactation. And as we can see here, if cows had metritis in one lactation, then they were much more likely to develop metritis in a subsequent lactation. So cows that had metritis in one lactation had an incidence of metritis in the subsequent lactation of 20%. And cows that did not have metritis had the incidence of metritis in the subsequent lactation of around 10%. So we see a doubling of the chance of these cows develop metritis in the subsequent lactation. So here our conclusion from this is that having metritis does not protect the cow. Actually, there's something to those cows, something intrinsic to those cows that makes them more likely to develop metritis. And we see that effect even after adjusting for other things that affect the chance of this cow developing metritis, such as induced parturition, having dystocia in that lactation, having twins in that lactation, stillbirth, retained placenta, or clinical ketosis. We still see that large significant effect of having metritis in the previous lactation. So what do we know about the genetic components of uterine disease such as metritis and clinical metritis? Actually very little. We have done some studies and one of the things that we looked at is the influence of this modification, this SNP, the single nucleotide polymorphism, which is a change in the genetic code for a certain gene. And specifically, we were looking at the change in this receptor for this molecule called IL-8, which is a molecule that actually called the troops in, the neutrophils, the leukocytes, to fight an infection. For example, metritis or clinical metritis that happens in the uterus. So we thought that that might be important for the development of metritis, because this SNP, this SNP in the IL-8 receptor had been shown to be associated with mastitis. So we thought if it is predisposing factor to mastitis, it is probably affecting metritis as well. But to our surprise, we found no association between this modification in this genetic code and the chance of the cow developing metritis or clinical metritis. So although we think that the immune system is working in a similar way all over your body, here we see that there are differences depending on the tissue or the organ, such as the mammary gland or the uterus. So the same SNP, the same change did not cause the same effect. Well, then we moved on. We looked at this, what it's called toll-like receptor, which is what parts of the microbes bind to stimulate an immune response. Again, we looked at several of these SNPs and several of these toll-like receptors that bind to different microbes, and we did not see major effects on metritis or clinical endometritis. And the last thing that we know about the genetic components of metritis is that it has low heritability. And what that means is that there is a low percent of the variation that is explained by genetics. That tells us that there are environmental factors that are contributing the most to the instance of metritis in dairy cows. Although the heritability is low, between 7 and 10%, over several generations, we can make a genetic improvement. So this is what we wanted to do here. This is data from our geneticist here at University of Florida, Dr. Peña Garicano. So we collected data from one of our clients, with a large dairy herd. So here we collected phenotypes, so the incidence of disease from 28,000 health records from 14,000 hosting cows across several lactations. And we categorize those cows as either having metritis or never developing metritis. So either had metritis in any of the lactations that were available or they never had a metritis. And here they used the 60,000 SNP chip, they clarified from Zoetis to genotype these cattle. So we have a genotype of 8,000 animals. So in this procedure, we combine the information of the genotype from 8,000 animals with the pedigree information from those 28,000 animals. And we can, the geneticist can make predictions whether there is a genetic component to this disease, metritis. Again, just showing here the numbers, 8,000 animals with genotype and 28,000 animals with pedigree information. And we're looking here in this graph, the results that we find here, the percent of genetic variation. So when you see these spikes that goes very high, that means that there is a gene or a set in that chromosome that explains a large proportion of variation. So those are the ones that we want to focus on. So here on the bottom, the x-axis, we have all the chromosomes from the cow, from chromosome from 1 to 29, excluding the sex chromosome. So the cow has 30 pairs of chromosomes. So we're not interested in the sex chromosome, we're just interested in the other ones. So here there were a couple of these spikes of interest, and here these letters here indicate genes that are in that region of that chromosome, and we'll see what those are. And here at the bottom, on the bottom left, we have the heritability that we found on this study, and it was in line with the previous literature of a heritability of around 9%. Again, low, but again, we can make progress over several generations. So let's take a look and see what those genes actually are. What do they do? What are they responsible for? And what they have been associated with. So these are the putative candidate regions and genes. So some were associated with inflammation, and that's interesting because of course, metritis is the inflammation of the uterus. But this gene was not really involved in what we believe is the most important thing for uterine disease, which is the innate immune response, the immune response in which neutrophils are involved with. This was more involved with chronic inflammation, such as Crohn's disease or colorectal cancer, never being associated with infectious disease in cattle. So we were a little bit disappointed with that. The other genes that we found were mostly involved in cell cycle proliferation and death, which are also genes that are associated with cancer. So metritis is definitely not cancer. So we got, again, a little bit disappointed with that. But again, maybe there's information about these genes that we don't know. And all we need to know if they are linked to metritis, and we can select against those genes or the SNPs that predispose to metritis, and we can exclude them from the population. We can still select for improved fertility. Then after this study, we decided to take a step back and decided to look at one of the most important risk factors for metritis, which is clinical and subclinical hypocalcemia. This is one example only, but there are several studies in the literature that show a negative effect of clinical or subclinical hypocalcemia on the risk of developing metritis. So here, for example, we have here cows that were classified as having normal calcemia. Those are normal cows, or cows with subclinical hypocalcemia, and the incidence of puerperal metritis, which is metritis with a fever. So we see how here cows with normal calcemia, the incidence of metritis of 2.5%, very, very low. And cows with subclinical hypocalcemia with an incidence of metritis of 44.4%. Very high incidence of metritis, just showing the importance here of calcium, because calcium is actually what activates the immune cells. They activate them so they can fight off disease, they can phagocytize, and they can kill bacteria. Very, very important. Besides that, we saw an effect of parity. We usually see primiparous having higher incidence of metritis than multiparous, and also a mild effects of risk group. So the low risk group were cows that did not have risk factors such as dystocia, twins, and stillbirths, and cows with high risk factors were cows that had those predisposing factors such as dystocia, twin, stillbirth, and retained placenta. But even then, after having adjusted for calcium, the effect of hypocalcemia, the effect of risk group, although it's large, was just a tendency, not a significant effect. What about the genetics? So what did we find here? Again, we see regions in the chromosomes that have these spikes, which means that they explain a large percentage of the variation in the incidence of subclinical, or in this case, clinical hypocalcemia, milk fever, in this case, milk fever. So here we have spikes in several chromosomes. Here we have in chromosome 2, one in chromosome 3, one in chromosome 6, one in chromosome 14, one in chromosome 23. And what do those genes are involved with? What do they do? So what do these genes actually do? All of them are actually involved in vitamin D metabolism. And we know that vitamin D is very important for calcium homeostasis, for maintaining normal concentrations of calcium in blood. So for example here, vitamin D first has to be produced in the skin through the effect of the sun. So we need sun exposure in order to manufacture, in order to make vitamin D. But this vitamin D is in the inactive form. And the inactive form of vitamin D needs to be transformed first in the liver, then in the kidneys, to become the active form of vitamin D, which is called calcitriol. And these genes that we found, two of those genes that we found that were significant in our GWAS analysis, that's what they do. They're involved in this hydroxylation, which is a transformation of this inactive form of vitamin D into the active form of vitamin D. So after the active form of vitamin D is produced, action needs to be transported to the target sites, such as intestines, where calcium is absorbed, kidneys, where calcium is reabsorbed, so we don't lose too much calcium into the urine. And if needed, calcium needs to be reabsorbed from bone. So bone reabsorption is also an important mechanism for maintaining normal calcium concentrations. So this GC gene is involved in vitamin D transportation, so vitamin D production of this vitamin D binding protein. And this vitamin D is transported to the final target, which is the DNA itself. That's where it binds to its receptor and stimulate the production and its effects in calcium homeostasis. And we have two other genes that are involved in this final step, so it can actually have the effect that we want, which is to maintain normal calcium concentrations in blood. So here the results for hypocalcemia in our GWAS analysis were much more exciting than for metritis. And we know that hypocalcemia is an important, a very significant risk factor for the development of metritis in dairy cows. So with that, well, just some take-home points. Uterine diseases are highly prevalent and deleterious to fertility. It affects both conception rate and also pregnancy loss. Genetic variation does exist for metritis, but heritability is low. So genetic progress will be slow, although it's possible, and we should be emphasized. So our GWAS analysis produced a list of putative candidate regions and genes that were involved in inflammation and cell cycle proliferation and death. And we need to do a little bit more digging to see what exactly those genes are doing that it predisposes the cows to metritis. In the case of milk fever, we saw that major candidate genes are closely related to vitamin D metabolism, which is important for maintenance of normal calcium concentration in blood. And also those have biological functions as well. So here we have a nice story to tell, and we have good targets to improve calcium metabolism and decrease, hopefully, the incidence of clinical and subclinical hypocalcemia in dairy cows, and hopefully they'll improve health as well, including uterine health. With that, I'll thank you very much.